Okay, transport systems in vertebrates. Today we're talking about the circulatory system, the excretory system and the respiratory system and how they work together to get the nutrients our cells need to where they're needed. So what we already know is that materials need to be moved around the body in order to be either used by another cell or moved from the body, as in the case of wastes. We also know that there's a hierarchy in the body, and you would remember from class that I showed you this beautiful pyramid here, where we start with the largest thing at the bottom, the organism, and we get to the smallest part of that organism, the atoms. Physicists would probably argue with me that they're not the smallest, but in our case, atoms are the smallest. So the organism is made up of systems. Systems are made up of organs. Organs are made up of tissues. Okay. Tissues are collections of cells who work together. Cells are made up of organelles. Organelles are made up of those biomacromolecules we talked about earlier in this unit, and those biomacromolecules are made up of monomers, little subunits that form the long chains of biomacromolecules, and those molecules are made out of atoms. Cells need molecules and biomacromolecules to function, and because we're consumers, animals need to digest the biomacromolecules made by other organisms. So during digestion, we break down tissues and organs of other animals into small molecules that can be used by our cells. And you would remember from when we talked about digestion, we were talking about carbohydrates, amino acids, lipids and fats, nucleic acids, and even some smaller products of digestion, water and minerals and vitamins. And all of those are necessary for our cells to function properly, so we need to get them there. And we use a system, a transport system, to get them from the digestive system to the cell that needs them. That transport system is the circulatory system. Every cell in the animal body has a capillary nearby, and these capillaries are the smallest of all the blood vessels in our body. They're only a cell thick and just have enough space for one red blood cell to fit through at any one time. So they all go through single file. The circulatory system is where nutrients from the digested food can pass into that capillary via either osmosis with water or diffusion and transported to where they're needed. So inside the villi of the intestine are a whole heap of capillaries that are collecting those nutrients from our intestines. The bloods then move through the body by a closed circulatory system in vertebrates, which means that we use blood tubes rather than having all the blood just oozing around our body, around all of our cells. Insects have an open system, but vertebrates use a closed system. In the middle of the circulatory system is a double pump heart, and in humans, that's only in humans, which is where the blood from one side doesn't meet the other side. It has to go through the lungs first before it can be used. So it's like two pumps working at the exact same time. As you'll see in a moment, not all organisms have a double pump heart. Now, the other parts of the circulatory system that we talk about are the veins and the arteries. And the veins carry blood back to the heart, and usually they carry deoxygenated blood. That's blood that doesn't have oxygen in their red blood cells. So we usually show that as blue because they're actually really high in carbon dioxide as well, and the bicarbonate makes the blood look a little bit bluish. Now the arteries are the blood tubes that carry the blood from the heart, and we usually show those in red in diagrams because they carry oxygenated blood. And when oxygen binds the hemoglobin in the red blood cell, it turns a very vibrant red colour. You can see that there are a couple of veins that carry oxygenated blood, and they come from the lungs back to the heart, and they're called pulmonary arteries. And there is also a set of veins that go from the lungs to the heart, and they carry oxygenated blood. They're the only two exceptions in this case. If we remember where they're coming and going from, it helps us remember whether they'll be oxygenated or deoxygenated, because of course oxygen exchange happens in the lungs. So the arteries, there are a couple of different types of arteries. The very first one that comes off the top of the heart is called the aorta, and it has a really thick wall around it with lots and lots of muscle layers, and that's to protect it from being split, because when the blood is forced out of that left ventricle, it's under really high pressure, and if it wasn't a thick enough wall of your aorta, it would split and rupture and the blood would spill out of those blood tubes, and that would kill a person. So it has to have a really thick wall. The aorta then splits down into some smaller blood vessels called arteries, and these are still fairly big and they're still very thick walled because the heart is still pumping, and so they're still under pressure. So you can see the cross section here of an artery, and the muscular layers of this artery, and you'll see in a moment that a vein looks quite different. Now the arteries branch more and more, just like the highways in our streets. You often go from a major arterial road named after arteries. The major arterial road often has branches coming off it, which are also relatively major, like major roads, and they would be like the arterials, and then they would split off into even smaller roads that go to each house, and those little streets would be more like capillaries. So the aorta branches off into arteries, the arteries branch off into arterioles, and those arterioles branch off into capillaries to visit each and every cell. The capillaries return the blood back from the heart after visiting those cells and dropping off those nutrients by merging to form larger tubules called venules. Those venules then merge and form larger tubes called veins, and the veins then join the even larger veins to be called vena cava. And there's the two largest veins, the ones that empty into our heart, are called the superior vena cava and the inferior vena cava. Superior is easy to remember because if you're superior, you're up on top, and if you're inferior, then you're on the bottom. So the superior vena cava brings the blood back from the head, and the inferior vena cava brings the blood back from everything below the heart. So your legs and your digestive system would all come back from the inferior vena cava. Now, because the veins work against gravity and the blood has already slowed its flow as it went through all those capillaries, the wall of the vein is much thinner, and it also has to have valves to help it travel back up through our body. So as the blood goes up, the valve closes and stops the blood flowing back down into the feet again, or into the bottom of your body. If these valves are faulty, you can end up with enlarged veins called varicose veins. Okay, so once again, just showing how these branch off. So you can see that you've got this major arterial road here, the artery, that branches off into an arteriole. We're getting smaller and smaller. We're starting to be able to swap off the nutrients, pick up wastes, drop off oxygen, pick up carbon dioxide. We go into those capillaries. Now gas exchange and nutrient exchange is happening really, really well. We're visiting lots and lots of different cells, and we're now ready to start heading back toward the heart. So we form venules, and these venules form veins, and those veins form the vena cava that go back to our heart. So not all vertebrates are the same as humans, as I said before. When we're talking about mammals, crocodiles, birds, we are talking about a double loop circulatory system that goes through the lungs and has two separate parts of the heart. So it actually has two separate atria and two separate ventricles, and they're completely divided. So there's no cross flow. There's no mixing of the blood going from one side or the other. And this is quite a complex or evolved heart. So it shows us that evolution, if you're thinking about evolution here, what we're saying is that these animals, crocodiles, birds and mammals developed a lot later than most reptiles and fish because they have quite immature or unevolved hearts. But as you see in a moment, you'll see that they have some similarities. So most reptiles, other than the crocodiles, have a double loop circulatory system, so it still goes through lungs. They have developed lungs at this point, but there is a hole or a partial opening in their heart. So there is mixing of blood between the left-hand side and the right-hand side. This is fairly inefficient, which is why we've later developed to have a closed wall, because with that mixing, you're getting less oxygen to your cells and more carbon dioxide to your cells. Fish are even more immature. They have a single loop system. It doesn't go through twice. It simply goes from gill capillaries through the heart, back through some more gill capillaries to the body, and back again. So it's just like having your two lungs still, but the heart's a one-way trip. It just keeps pushing it right around in one go, and it's very simple. So the human heart, as I said, it's a double pump, and there's this bridge, this muscle wall in between that stops the blood going between the left and the right side. You can see we're using that same colour coding system here for blue, where there's deoxygenated blood, and red, where there's oxygenated blood. If you imagine that this heart is sitting on your body, so if you imagine that the left-hand side of this heart, where it says left atrium and left ventricle, is pointing to your left hand, and the right atrium and right-hand side is on your right-hand side. That will just help you orientate the heart to you. You can see here that we have the superior vena cava entering at the top, and here's the inferior vena cava, and that opens up just in, two superior vena cava there and the inferior one just in here. So when we do a heart dissection, we're hoping to find two holes into the right atrium. The right atrium is where the blood first collects, and once we've got enough blood in here and enough pressure, the heart beats, the valve here, the tricuspid valve, opens and the blood flows into the right ventricle. That blood then pulls in here very quickly into the right ventricle. Next heartbeat, up it goes through the pulmonary valve, and this now takes the blood to each lung. Once the blood's been to the lung and it's picked up oxygen, it returns via the pulmonary veins, and you can see that there are two from each side here coming into the left atrium. So we can tell the left atrium because it has these four openings in it, although they are quite small. The blood then, once it's pooled in the left atrium, flows in on the next heartbeat down to the left ventricle through the mitral valve, and then on the following heartbeat gets squeezed out through the aortic valve and out the aorta, going up to the head or down to the body. As you can see with this, the right atrium has the thinnest wall in your heart, and that's because it's getting that very low pressure blood dripping in back through the vena cava. There isn't much that pulse left once the blood's been right through your body. However, the right ventricle needs to have a thicker muscle wall because it has to have enough force to squeeze that blood up through to your lungs. Again, once it's gone through your lungs and all those capillaries, the pressure's fairly low, so the left atrium only has a very thin wall as well. But once you're in that left ventricle, your heart now has to have enough force to pump that blood all the way around your body again, which is a much bigger trick. So the left ventricle's wall is much, much thicker than the right ventricle's. Okay, so what happened when the blood was up at your lung? Well, this is where the respiratory system kicks in. So these two systems are working together to make this organism work. So when we're talking about the respiratory system, we're talking about the gas exchange that needs to occur because our cells need the oxygen for cellular respiration, and once cellular respiration's occurred, we need to get rid of carbon dioxide. This exchange happens in the respiratory system in these little tiny bubbles at the bottom of your lung called the alveoli. You can see here that there's a capillary network wrapping around those alveoli, and that's where that gas exchange happens. We're talking about moving these gases through basically two cell layers, one of the alveoli and one of the capillary. You can see that the lungs work very similarly to the circulatory system. The body loves this system. One pipe in called the trachea, and then we have bronchi, which are still bigger pipes, and then we have secondary bronchi, and then we have bronchioles, and then right down the bottom we have these alveoli. So we're getting smaller and smaller and smaller, ever increasing our surface area of our lungs. And by increasing the surface area, it means that we can exchange gases much more efficiently. You can even see the bubble shape of the alveoli means that we have the maximum amount of surface area for the space that we've been given, and this means that our lungs can be very compact even though they're doing a lot of work. So basically, that pulmonary veins transported that deoxygenated blood from the heart to the lungs, and the blood is very high in CO2 and low in O2. That CO2 needs to move through the capillary wall and into the alveoli via diffusion, and at the same time the oxygen moves through the alveoli of the lung and into the capillary, so that oxygenated blood can return to the heart via the pulmonary artery. So how does this happen? Well, if this circle is our alveoli, inside it when we breathe in, so we're going to breathe in here in the alveoli will be lots and lots of oxygen. Lots of oxygen. Much more than is in the capillary. Now, of course, there's still oxygen in the capillary, but because there's more inside the alveoli, that oxygen is going to come out and attach itself to the hemoglobin in the center of the red blood cell. So by the time the blood has flowed around, there is a lot more oxygen in it. So now we've got lots of O2 attached to our red blood cells. I apologise for the colours, it's a bit difficult. So that oxygen is going to come out and attach itself to the hemoglobin. At the exact same time, something else is happening. Inside our blood, we have carbon dioxide, lots and lots of carbon dioxide in our capillaries because it's being produced by cellular respiration, much more than was in our alveoli, in our breathe in. So here we've got a little bit of carbon dioxide inside our alveoli and lots of carbon dioxide in the capillary. So we know that the concentration gradient is higher. It will move from high to low here. So if we're going from the high concentration to the low concentration of CO2. So this CO2 is going to rush into the alveoli and we can breathe it out. So I'm going to breathe it out. So both of these things are happening at the exact same time. So by the time we are ready to leave, the blood's ready to leave the capillary or the lungs and start joining back up into the pulmonary artery, we now have blood that has very few CO2s in it and an awful lot of oxygen. Of course, we still have oxygen in our breath out, and that's because, well, we know that because otherwise respiration, when we do CPR, would not work. Our breathe out would just hurt the person more, but because there's oxygen in there still, just at a lot lower concentration, we can try and save someone's life by breathing into their chest. Okay. So the last system we need to look at, and the one that we haven't gone into much detail with yet, is the excretory system. So the excretory system is where those wastes are removed, and there's actually 3 different ways that we do that, and one of them was the respiratory system, when we get rid of our carbon dioxide that we don't need anymore. Another way we get rid of waste is through sweating, and through sweating we can get rid of salt. And if you've ever had licked your skin once you've been sweating or running, you'd notice it was quite salty. So sweating gets rid of some excess salt, and our urinary system is how we get rid of salts and nitrogenous waste that we don't need anymore as well. So our urinary system is talking about the kidney, the ureters, the bladder, and the urethra, how we filter and get rid of our blood. So if this is the cross-section of the kidney here, you'll see that there's this section in the middle here called the hilum, and it's usually very smooth, and it's like an indentation. It's what gives the kidney its beautiful shape. That hilum is where the renal artery and the renal vein come in and go out. So you can notice that these blood vessels are quite big, because we need a lot of blood to go into this kidney to be able to filter it. The rest of the kidney is where that filtering happens. So out on the outside here on the cortex is where those blood vessels will be sitting, and in the medulla, the pink section, is where our tubules and our loops of Henle will be, our nephrons. So if I just draw in some capillaries in here, there's our capillaries. If I just grab another pencil here, around this glomerulus, we have a Bowman's capsule, which is like a little cup shape, and then this goes down into the loop of Henle, and then we have the collecting duct then would empty out into the renal pelvis. The urine collects in here in the renal pelvis, coming from all those different little nephron subunits, and then that urine will pour down the ureter going to the bladder. So here's another diagram of the urinary system. So as I said, we've got the kidneys, we've got the renal artery and the renal vein, and we have our ureters going down toward our bladder, and then the bladder, once it's full, that muscular bag stretches and a signal goes up to your brain to say, hey, it's time to go to the toilet now, and a sphincter is open just at the end of the bladder, and the urine will fall out through the urethra. In the women, the urethra opens out just at the front of the vagina, and in men, that urethra opens out at the end of the penis. So what is urine? Well, mostly it's water. The more water you drink during the day, the more water will be passed out through your urine, and the paler your urine will be because it'll be less concentrated. If you're not drinking enough water, then your urine will become more concentrated, and that's why there's this range between 95 and 97% water, because the more water you're drinking, the more water will be a part of your urine. Floating in that water are some solutes, some solids called urea, creatinine, ammonia, uric acid, and some salts. Now, there were three terms that we've been using when we talk about osmosis and solutions: hypertonic, hypertonic, and isotonic. And we also use these to describe the urine that's produced. Basically, tonic means the fluid, and hypo means low, and hyper means high, and iso means the same. So hypertonic urine is where the urine has a lower concentration of salts in it than the body fluids of that animal. And this happens in freshwater fish and amphibians, where we need to remove an awful lot of water, because a lot of water comes into the fish and that needs to be removed. Hypertonic urine means that we have a higher concentration of solutes in the urine than we do in the animal's body fluids, and this is mammals like us. So we have a lot more concentrated urine than freshwater fish and amphibians, because we have less water and we need to try and conserve that water. So we try and save it by producing more concentrated urine. Of course, we don't want it too concentrated, because otherwise that urine would become toxic to us. Now, isotonic urine means that it's the same concentration of salts, and this happens in marine fish where they live in quite a salty environment. So their water can be conserved at about the same rate, because it is water and salt that they're floating in. So their body fluids are quite salty and watery, and so their urine is about the same. Okay, so I started talking about these nephrons, these little blood filters, the little urine producing units in our kidneys. So they actually do start out in the cortex, out on the outside, and they move into the medulla. Okay, they start off with this double-membraned cup-shaped structure called a Bowman's capsule. And this Bowman capsule has all the blood vessels inside it from those capillaries, from the renal artery and the renal vein. So this is where all of the solutes and some of the water will transfer from the blood vessel into the nephron. So the nephron's basically like a giant big tube, and we have these tubules on this side and on the other side. On the side closest to the capillary, we call it the proximal tubule, and on the other side we call it the distal, or distant, I suppose you could remember it as, the distal tubule. And in between we have this thinner part called the loop of Henle. Now, the blood vessel net we called the glomerulus. Okay, so how does the urine actually form? Well, pretty much everything except for the blood cells, large proteins, plasma and platelets are small enough to get into the nephron. But the blood cells, proteins, plasma and platelets are way too large to get out of the capillary, so they stay in. Unless the person's quite sick, and with things like diabetes, those blood vessels become a lot more leaky, and the nephron will actually take in some glucose and some protein, and that will show up on a urine test, because urine shouldn't have glucose and protein in it. So it's a quick and easy way of working out whether a person has diabetes or not. So those small solutes, things like salts, bicarbonate, hydrogen ions, urea, they all pass into that nephron. And part of the time in the proximal tubule, we try and reabsorb things that we might be able to use somewhere else. So we try and conserve things like salt, bicarbonates, and even some water. So we don't want too much of our water being lost through the production of urine. So we try and save some of that, and that gets reabsorbed in the proximal tubule. At the same time, there's capillaries wrapped right around this tubule, and sometimes some drugs and toxins will also get placed into this tubule as well. So active transport's actually used to try and conserve or reabsorb some of those nutrients and ions, and of course the water just flows back with them to balance that concentration gradient. Now in the loop of Henle, that's where we're trying to conserve salt and also water. So we're able to passively transport the water out, but we also want to actively transport out things like salt, especially as we get into that distal tubule. It's like our last chance to try and capture back some of those things that we want to keep before it goes into the collection canal and off to the ureter, where it's going to be placed in the bladder, collected, and then urinated out. So now that that urine's filtered, it starts in this renal pelvis in here, and it kind of collects in there and then dribbles down through the ureter. So this happens all day, and you've got two kidneys, so you've got two ureters, and this fills up the bladder. So the bladder gets full, and when it says, ouch, it hurts now, the nerves around the bladder send a message up to your brain saying, quick, head to the toilet. And once you're there, a sphincter at the bottom of the bladder will open up. A sphincter is just basically a little ring of muscle that opens up, and the urine can come out the urethra, as I said before. So next week we're going to be doing some dissections of the heart and the kidney, and I've just given you a few images of what they look like right now. So these are, you can see the blood vessels at the top of the heart, these large, thick-walled blood vessels. Sometimes they're cut off, so they can be hard to see. This heart's been opened up. You can see here that you've got the right atrium and the valve, and then the right ventricle, and you can see how much thinner the right ventricle's wall is to the left ventricle over here. So then you have the left ventricle where the blood's now come back from the lungs, another valve, and you can see all the little beautiful heartstrings in there as well. And then down the bottom here we have that left ventricle before we go off through the aorta and out to the rest of the body. Over here we have the kidney, and you can see that the renal cortex is that brown section. And then inside the renal cortex are the renal pyramids, which form the medulla. So they look like little pyramid-shaped, almost like balls inside there. And you'll see that there's these white tubes in here as well that are forming those ducts that are collecting back that urine that's been produced by the nephrons. So the nephrons do start out here in the cortex, that's where all those blood vessels are, and then go down into the medulla and then into the renal pelvis by the collecting duct. Where you see the renal pelvis, you'll see that the ureter is connected to that, and that will head off to the bladder. So when we're doing this experiment, you need to remember the scientific diagram checklist. Make sure your diagrams are an outline only, and we usually use lead pencil to do this. We don't want to shade and we don't want to use colours, and one of the few exceptions for that is that we will occasionally use red and blue colours to show the oxygenated and deoxygenated parts of the heart, where they're carrying deoxygenated or oxygenated blood. The other thing we have to make sure is that we label them. So we want to draw our lines with a ruler, and we want to make sure we've labelled every visible feature we can find. We want to make sure there's a title, and we also want to make sure that they're big enough. Make sure they're at least 5 by 5 centimetres big, but really half a page or even a whole page would be more appropriate so that you can really show that detail. Take up all the available space you've got when you're doing a scientific diagram. Well, I hope that's explained transport a little bit more. If you have any questions, I'll answer those in class. Thanks.